good morning, everyone. So for those of you who wrote talking points about the arsenic article, please post one of your talking points into the chat room. We will be talking about the article briefly in today's discussion. So I'm going to give everyone about a minute to be able to do so, for those, again, who did write the talking point about the arsenic article. And while you're doing that, I would like everyone to just briefly, if you haven't done so already, to look at this YouTube video, which summarizes the article that we were talking about with regards to lithium. All right, so I want everyone to take a moment to look at this video, and then we'll continue on our discussion from last time.
The only way that you can make a therapy better is to understand how it was working to begin with. I'm Evan Snyder, and I work at SBP, and I am the director of the Center for Stem Cells in Regenerative Medicine. Bipolar disease, to the surprise of many, is a highly prevalent, highly lethal, highly incapacitating disease. It's the 6th leading cause of disability in the world. About 1/3 of patients will respond to lithium if they have bipolar disease, but nobody knows why. We found lithium's molecular target, did mapping upstream and downstream of the target, and mapped out what we like to call the lithium response pathway. Well, we found out that this pathway is absolutely critical for how nerve cells communicate with each other. With this kind of knowledge in hand, we know precisely what targets we would like other drugs to hit that are probably more precise than lithium, safer than lithium, with fewer side effects than lithium. If we find a drug that already exists, that can be screened out of a whole library because it's changed this pathway, that definitely accelerates progress to the clinic, and we could probably be in clinical trials in a year or 2.
Okay, so I wanted you guys to watch that. So I had asked you to click on it and watch it, but I had actually sent it to you last week for you guys to be able to watch it. So one of the things that I wanted to focus on is to emphasize the idea that whenever you're reading an article is to understand the big picture. Because I remember one of you posted a comment about the readability of an article, and part of what would help to contextualize an article is to get a little sense of, who are the authors, where was this work done. So this was a video that had been prepared by the professor who was the lead scientist on the manuscript. That doesn't mean that he necessarily did the experiments, as you would know. And so the key thing is to get the big picture, and then in having that at hand, when you start to read the full article, you get a better perspective of what they're trying to achieve. All right, so I'm going to go ahead and start projecting from where we were last time. All right, so again, an emphasis on participation being a big component of this course. I did see last time that people did not listen to my instructions about posting their talking points of people who actually had done talking points for the lithium article. So again, if you do not participate, that will affect your grade in the course. That is a big component. So please follow instructions whenever I ask you to post, because the idea is that if either you ask questions or you provide commentary, through the chat or directly getting onto your microphone. All right, so that's something that I would like people to follow along as far as the request when it comes to these talking points. So in our last discussion, this is where we were. The focus was on a proteomics approach to try to identify what proteins are potentially being affected by the treatment of lithium in these particular cells that were basically modeling lithium-responsive bipolar disorder. And so in this analysis, what was identified was actually something really critical, that this protein that was identified in the previous study, the CRMP2 protein, well, the actual expression of that protein wasn't really being affected by the concentration of the inhibitor. But what was being affected was a critical amino acid that was getting phosphorylated. And so what you can see as far as the use of this inhibitor is that the dose response, the higher the concentration, the lower the amount of that phosphorylation event. So then this was actually really important because it showed a correlation between what was going on as far as the potential relationship between that enzyme, the GSK3B, and the phosphorylation of the CRMP2 protein. Now the question was, what role was the lithium playing here? And so in this study, they looked at dosing with lithium and also other cations, so sodium and magnesium. So I mentioned this last time. And what was observed was that in the presence of lithium, only those experiments that did have lithium present was the level of the phosphorylated CRMP at that T514 was that actually getting inhibited. So you see a decreased amount of that protein. Again, you don't necessarily see an effect in terms of the levels of the actual protein itself. So that's something that's very important to get this idea of what is it that the lithium is actually doing and how it is actually distinct from other cations. Remember, lithium takes advantage of magnesium and also sodium transport mechanisms in order to be able to distribute in the body. So even still, even though it has comparable ionic radii as magnesium, it has a very distinct feature here. And so we can see there's a correlation. This other study was really important to get a sense of what exactly is being affected. And as you can see, it's a sort of a complex network of things getting affected. All right, so what was being examined here was the effect of the lithium and also that GSK3B inhibitor. So what we can see from that first arrow, that in the presence of lithium, okay, there is an increase in a phosphorylated form of the GSK3B enzyme. So that particular increase is of a phosphorylation site of S9. And so, as they report in the manuscript, this particular form of the enzyme is attributed to be an inactive form of the enzyme. Then, what was also observed was a reduction in the levels, restoring the association of the CRMP2 with tubulin. All right, so that was something that was done in a corollary experiment by co-immunoprecipitation. So basically, the administration of the lithium at that 10 millimolar concentration helped to basically increase an inactive form of the enzyme and therefore decrease the phosphorylation of the CRMP2, keeping that protein active. So things that are not shown in this particular set of data, but that appear to be correlated with what has been reported here, was that the lithium increases the levels of the active AKT. So basically, that is the enzyme that is attributed to the phosphorylation of the GSK3B enzyme. Right, so lithium is affecting apparently the phosphorylation levels of different proteins, and that network of effect is what's impacting the activity of the CRMP2 protein. Also, the lithium relieves inhibition of protein phosphatase 2A, which dephosphorylates CRMP2. Right, so it seems that the lithium is doing quite a bit in terms of regulating and trying to increase the amount of CRMP2, the active form. All right, so here I just provide you a summary of what I just mentioned, but I want to draw your attention into one key thing here. And so it's this point that I make right here. There is an abnormally high CRMP2 phosphorylated T514 with respect to just the protein itself, CRMP2. So that ratio of the phosphorylated form of the protein versus just the protein in general appears to be disease specific. Okay. However, the GSK3B inactive form are not lower in the lithium responsive BPD cells, indicative of the condition not being just a GSK3B protein problem. So with this collection of results and findings, the authors made the hypothesis that this high ratio, phosphorylated protein versus just the protein itself, is a molecular marker of lithium-responsive bipolar disorder condition. So then the next thing that they wanted to examine was a correlation of what was observed with those particular cells, those neural cell lines, with in vivo mice models. Okay, so what they did was to work with a wild-type mice with this background, the bulb CJ and then the C57BL6J, typically referred to as black mice. So this background, they were treated with and without lithium. And so the idea was to see, is there a correlation with what was observed with those neurons with what is observed in these particular mice? And so what we can see in this data over here is that with the presence of lithium, you do have this increase in the amount of, I guess you can call this the ratio of inactive GSK3B protein versus just the protein in general. But then also in the presence of the lithium, you observe a decrease in the ratio of the phosphorylated CRMP2 protein versus just the protein total. All right, so there does appear to be a correlation between what was observed with those neurons with what was being observed in vivo in this particular mice model. Another really important thing was observed, another in vivo experiment using a different mice model. Here, experiments were done with the CRMP2 knockout mice. All right, so what that means is that basically the gene expressing that protein has been knocked out, so therefore you are not going to have that protein present. So what was being observed in this experiment was the ramification of not having that particular protein present versus wild-type mice, right? So you have data for wild-type versus the knockout. Okay, so these are the different things that were observed. All right, so in those knockout mice, they did have normal bodies and brains. Okay, so as far as like physiologically, any effects, none were observed on that level. But then when you look deeper, these were the things that were observed. There was a unique dendrite embarrassy. Embarrassy means an abnormality. So there was loss of spine density. So spine density starts to get into the atrophy. You have loss of, I guess, to an extent, mass. Increased bifurcation. You also had the mice exhibiting behaviors, for instance, being hyperactive, over anxious under stress, less social, and they were not responsive to lithium treatment. All right, so this was observed in those mice in which that particular protein had been knocked out. And so, you know, corollary findings with this being observed in the wild-type mice, that the levels of that phosphorylated protein not detectable in the dendritic spines, and that lithium inhibition induces increases in spine density and area. So the data suggests that the CRMP2 function is pivotal for proper dendritic branching and spine organization in vivo. And so one additional study was done, and this is with samples collected from humans. And so here I provide some information with regards to the selection of patients that were utilized post-mortem, so after death, for this particular analysis. And so this was the data that was observed. And so shown are data for the dorsolateral prefrontal cortex of BPD patients. All right, so the main observation, the data suggests that excessively elevated CRP2 phosphorylated protein versus the protein total ratios are associated with the following attributes: diminished dendritic spine morphology, and so both of which are improved by lithium administration. So here you can see how they're able to observe this. So you have the measurements of the ratio of the phosphorylated protein versus the total protein, and then what is observed in terms of the presence and absence of lithium. Okay, and so that basically gave an indication of the correlation that was established with the neurons, with the mice models, and now with these human tissue. I'm actually going to skip over this just to make mention that calcium plays a very significant role in terms of signaling. So there does appear to be some impact in terms of what lithium is doing in terms of the transport, the signaling of calcium. But I think this is a little bit more complex than we have the time to really discuss here. All right, so with that in mind then, I want to now go to and focus on the other article that I had you guys read. So here, what we're looking at with the lithium article, we were looking at something that is a big question mark as to whether or not that metal is essential, but certainly it's therapeutic. However, its therapeutic window, it's very, very, very narrow. And so as we were talking about last time, it's very easy to go in an excessive amount, leading to a lot of side effects that makes it very complicated to use that particular metal as an absolute treatment. So the objective of this study was really to try to identify pathways that could be drugged to deliver something that's more specific and not entail the high levels of toxicity because of that narrow, limited window of therapy. All right, so give me a minute. I'm going to go back to looking at the other article. Okay, so in this particular case study here, we're actually deviating from that list of this idea of metal homeostasis. What we're looking at here is how your body also has additional mechanisms that it can take advantage of to help protect the body from toxicity of metals. And so this was material from that article. The gut microbiome is required for full protection against acute arsenic toxicity in mouse models. And so I did see some people write about this idea of not liking the title being about full protection. All right, so that's a wording issue that I do think is important. The choice of words in your titles in terms of what you're trying to say and then what you actually demonstrate in the actual article. All right, so it appears that Jael was the only one who wrote a talking point for this article. So he was a brave soul and wrote his write-up for the lithium article. All right. So there's a few things to mention before we really go into this article. And it's the understanding of the microbiome in terms of really the complexity of it and how abundant we have microbes in our body and we're living with them. All right, so I'm just gonna state some of these fine points. For instance, we have 100 trillion symbiotic microbes live in and on every person and make up the human microbiota. 95% of our microbiota is located in the gastrointestinal tract. 150 to 1, the genes in your microbiome outnumber the genes in our genome by that ratio. Okay, you have 1.3 times more microbes than human cells. They have 5 to 1 viruses to bacteria in the gut microbiota. All right, so as you can see, we do survive with other organisms. And so part of this study was to assess, well, what potential contribution does the microbiome play, at least with respect to, protecting us or even metabolizing certain things that enter our bodies, in this case, a metalloid. So here are the overall objectives of this particular study. They basically want to get an understanding, or at least to some extent, some understanding of the regulation of arsenic toxicity in mammals. And so these points here, these are things that you can take from the article itself. I'm going to just get to the heart of the article so that we can move along. What they did to get a beginning understanding of this was that they used mice models to determine whether the human and mouse microbiome exasperates or protects the host from acute arsenic toxicity. And then finally, to examine the importance of a particular enzyme, this arsenic 3 methyltransferase, which is responsible for metabolizing arsenic. All right. So in terms of the approach, right? So we know that there's mice work that was done here, but what exactly was the approach that was taken to try to get some fundamental understanding of this correlation between the microbiome and arsenic regulation? So what they did was they perturbed the microbiome to measure how it affects arsenic excretion and bioaccumulation, to determine whether the arsenic 3-MT, that particular enzyme, and microbiome increase survival and thus play a protective role, and to determine whether human microbiome transferred to mice can provide host protection. Also examine whether donor-dependent variation affects protection, right? So that particular point goes into the concept that the human microbiome, right, is distinct. So each person has his own distinct microbiome. And so the question here was, well, can that have an effect when you do this this transfer to mice in terms of being able to potentially serve a protective role? So here I provide the list of mouse models that were used. I will caution you, when you're writing your assessments of these articles, be very careful about when you're talking about mice, because mice, you have to be specific about what mice model is being used, because I did see some language about these experiments were done with mice, but many of these experiments were done with particular mice for a reason. And so I do expect you guys to do the work of identifying, well, why was a particular mouse used, and what exactly is being, what is exactly being observed, or the bigger objective from the use of that mouse. And that's one thing. Another thing is there was some language issues of people confusing rats and mice. Those are two different things. And so be very careful about the language there as well. All right, so these are the mice models, the antibiotic treated. So basically, they use this broad spectrum antibiotic, and then they examine those mice under certain treatments. Transgenic, so genetic material transferred from another organism. So in this context, we were talking, at least in this article, they discussed using some fecal matter from humans and placing into mice. Also germ-free mice, and autobiotic mice. So these mice having only certain strains of bacteria. So it's very important to be able to understand what are the mice models and what their contributions to the work is going to be. All right. So with regards to the objectives, the very first one being perturbing the microbiome to measure how it affects arsenic excretion and bioaccumulation. So as far as the experimental overlay, I believe that this is something that's pretty straightforward to understand. So they use this black mice were either administered the broad spectrum antibiotic, or they were basically given a sham treatment. So nothing, just the background, the buffer that was used for 2 days prior to and throughout a 14-day exposure to different amounts of the inorganic sodium. And so here, 25, 10, 100 ppm, there was also points where they used 10 ppm. So the thing, I wrote inorganic sodium, and I saw many people also wrote inorganic sodium, but also what exactly is inorganic sodium? Sodium arsenate 5. So, you can look that up and see what that looks like. I repeated the same language as the paper, but really you guys can quickly look up what that species would be. So you get a sense of the speciation of what is that arsenic 5. So what they were trying to do was examine the content of arsenic in mice stool and also in organs. All right, so here I show you representative data. And so what we're looking at is the dose of the arsenic in the mice treated with the antibiotic, and then those that were not, right? And so you just get the corresponding number of mice per experiment. And then we're looking at here the arsenic in stool versus days of exposure. So as far as that set of data there, this suggests possibility for greater excretion in the mice that were not given antibiotics. So you can see that those that were, there's much less excretion of the arsenic. So what does that potentially mean? Well, that could potentially mean that the arsenic is staying in the body. And if arsenic is a highly toxic metalloid, that can have big detrimental effect on these particular animals, could lead to death. All right, so that's what's being observed here. The using that just very general antibiotic has a big impact in terms of the excretion of the arsenic. All right, so that starts to get into this idea of the metabolism of the metalloid. All right, then what about this data down here? Well, we're looking at distribution of the arsenic, again, under those two concentrations in different organs. So the liver, the spleen, the heart, the lung. All right, so the first thing I wanna ask you guys before I give it just an overview. So you see these bar graphs, but you also see above those bar graphs, you see these dots. What are they? What are those dots?
Outliers.
So Adriana says outliers. Anyone else? I'm not saying that Adriana is wrong. I'm just wondering, anyone else? Okay, so typically, I would say there is a bit of a move, although you don't see it in many papers, to try to move away from the use of bar graphs. And why is that? Because bar graphs can be pretty deceptive in terms of the range of the data, even if you include error bars. So oftentimes what people will do, you'll see some papers where they simply will report the actual raw numbers and no bar graphs. Here, there's a combination of both. All right, the combination of the raw data plus the bar graphs plus the deviations of the numbers. All right, so just to, as a point of reference, because you do see a move away from the use of bar graphs because it can be quite manipulative. All right, now what was observed here? So just by looking at the data, you can see where there is a report of a p-value. So p-values can be a little bit controversial in terms of what they actually mean. But people use them as a way of expressing statistical significance, and sometimes they kind of use it in a predatory way where they just see a small amount of change, but if they get a p-value that's lower than a certain amount, that means that definitely that's statistically significant, even if it doesn't really mean much physiologically speaking. But anyways, this number quite low suggests there is a statistical difference here. And so generally what one could say is that there is greater tissue accumulation in the Seth group with the 25 ppm dosage. I would argue not so much difference between the sham and the Seth group at the 100 ppm, okay? There is a difference here. If you look at this data, there's somewhat of a difference there, but there's quite a bit of overlap for the organisms. And that just could be that you just reach such a high amount of the metalloid that regardless of having the, whatever microbe is being affected by the antibiotic, it just is a threshold that's too high that you're just going to get a high accumulation of the metalloid. That would be my interpretation of that data. So the next objective, to determine whether that particular enzyme and microbiome increase survival and thus play a protective role. So as a point of reference, what does that enzyme do? It basically serves to biotransform the arsenic. It methylates it. So now you make it into a form that potentially can decrease its toxicity and it can be excreted. So what they did was they worked with mice that did not have that enzyme. And so these are known to have to experience a greater arsenic-induced cytotoxicity. These mice were examined to determine whether they would have greater sensitivity to arsenic treatment versus all type. Another thing that was done as far as this was examine the survival of the mice populations to 10, 25, and 100 ppm of the arsenic 5. So these are the populations that were examined. The AS3MT knockout treated with the antibiotic and without, and then the germ-free AS3MT knockout mice. All right, so here we get a general summary of the data. So some of the data that was presented. We're looking at percent survival versus days of exposure at different dosages, okay? And then for the bottom experiment, you see that there's a mixture of populations that are being examined here, where we're looking at the knockout mice without the treatment of the antibiotic, and then we're looking at knockout mice that are also germ-free at different dosages of the arsenic. All right, so what is generally observed from this data? I would say this is a pretty straightforward one, where the knockout mice are indeed more sensitive to arsenic exposure to wild type. This is actually not shown in this data, but that's something that was shown in other parts of the article. The data here support the debilitating effect of the combination of the absence of the enzyme and also the perturbation of the microbiome. So you have that combination, perturbation of microbiome or the absence of the germs in general. What I personally, and so if you want to look at it closely, you see the effect of survival, where you have less survival in these conditions, where you have the antibiotic present, versus you have longer survival when you don't have antibiotic present. So that's just the perspective. If you didn't see that in this data, looking at how the survival is getting affected under those different conditions. So obviously, longer the days of exposure, and then the percent of survival is an indicative of the effect, of the impact of these conditions. But one thing I did not see, and I looked closely, was a data set that showed inclusion of a germ-free wild type version to see what impact it would have. I mean, I think it's pretty logical what we can expect to see, but it's something that is always good to have this control. Another experiment that was done was just to verify that the antibiotic at the dosage that was being used was not contributing to the mortality of the mice. And so that's something that they were able to establish in this work. All right. Another really significant thing. So they were able to show, obviously, the effect of the use of that general antibiotic. They were able to show how not having germs available, so not having some amount of these microbes available to be able to process arsenic, have an effect on the survivability of the animals. But what if we did the following? Determine whether human microbiome transfer to mice can provide a host protection. So what was done was germ-free, the AS3MT knockout recipient mice received a fecal transplant from human donors prior to exposure to the arsenic. And so they had two populations of these, what they called humanized mice. The F0, so that's the mice which would receive that fecal transplant, and then the offspring, the F1. So what they were trying to do was examine the variability of mice survival based on different donor microbiome. And so these were the general findings that were observed. So this is the data of survival versus days of exposure. So the human mice exhibited percent survival comparable to native microbe containing mice, and the percent survival varied depending on the donor, indicative of difference in bacterial strains, which, as I already mentioned to you, the microbiome profile is unique to every person. So that's an expectation that that would be an expected result. Along with this was a identification of potential microbiome members. And here, obviously, it was a very select study. This was not meant to be a comprehensive thing where they were trying to identify all of the microbes that may be actually correlated. It was to get some insight and to try to validate one finding of the collection of data that they have. So they wanted to identify microbiome members that correlate with survival. And so they did this 16S RNA gene sequencing performed on stool samples from humanized mice at the outset of arsenic exposure and also at day 7. And so then the classification was determined by blasting the data set against the ribosomal database project. So they tried to determine gut bacteria that exhibit detoxification properties with survival modeling of the 100 most abundant operational taxonomic units, so OTUs. And so this is basically a representation of their data, where they're looking at different, in this context, we can refer to them as microbes, and how the heat map, so the amounts of these, depending on the donor, the human donor sample that was collected and placed into certain mice. So these black bars that you see here, they indicate the strains that were observed on day 0 and also day 7. So a negative coefficient indicates a protective association, while a positive coefficient, so we're looking at these numbers here, are associated with increased hazard. And so what they did with that information is they wanted to say, okay, so that's what was their general understanding of this data, this correlation between the presence of these organisms and potential associated with the regulation of arsenic treatment. What they wanted to do was then they selected one particular member and tried to see, well, or further studies, does that particular organism have an impact in terms of the responding to arsenic treatment? So they wanted to explore the protective capability of it. The combination of results from previous studies, including the already known information that this particular organism, this F. prausnitzii, as a probiotic, results from humanized mice, and the bacteria's ability to produce that actual AS3MT enzyme, that collection of knowledge led the research team to hypothesize that it provides protection against arsenic toxicity. So that was their hypothesis. And so what they did was, again, a survival study versus days of exposure. And so here they had a bit of a technical limitation where that particular organism did not colonize in the mice, and so it had to be co-colonized with E. coli. So it had to be in the presence of a bacterium, E. coli. But the mice with this F. prausnitzii experienced higher percent survival when treated with 25 ppm arsenic. All right, and so as you can see here, this data, there is with greater days of exposure, there is more percent survivability. So that indicates that that particular organism can contribute to survival, but that doesn't mean that it is the only one. And obviously, as I mentioned to you, it was not meant to be a comprehensive study. It was meant to use that sort of bioinformatics approach to then identify one representative that falls into that correlation of potential protective role, and then examining whether in vivo it does exhibit that ability. And according to this data, it does. All right, so with that in mind, let's see Giles' question. Will generally humans who lack that bacterium be at higher risk of the toxic arsenic, and is there any evidence to support this? So, that's a complex question to ask, in that there are so many different bacteria that it may be that there might be some level of effect, but there might be other microbes present that can probably balance out the loss of that. So that's something that would require, trying to do a systematic study, which would probably be quite difficult, to try to see what happens if you just isolate that one, remove it, and what will be the effect. So it probably isn't that would be enough to then make human, or to induce that humans are more likely to be susceptible to arsenic toxicity. I think it's a correlation of all these microbes interacting with one another that would probably be the key thing. So yeah, I would say probably not necessarily, but it obviously has some correlation with serving some sort of protective role in terms of the exposure to the arsenic. All right, any other questions with regards to this article? All right, so no question. I see that Christopher posted his talking point about the lithium article. Let's see. En el artículo discutido anteriormente se presenta un análisis de la vía de respuesta al litio para la reducción de la concentración de la forma fosforilada e inactiva de CRMPs como alternativa para la modulación del desarrollo de desorden bipolar, tomando en cuenta que la hiperfosforilación de las proteínas tau también conlleva daños axonales que resultan en una cascada de reacciones que conduce al desarrollo de Alzheimer's. ¿Existirá alguna alternativa para establecer una terapia basada en la vía de respuesta al litio para retrasar la neurodegeneración? So again, that's a complex question, but you do see that correlation, right? That the lithium is having an effect on the phosphorylation of the proteins. And so he's going back to, and I do like that drawing connections, going back to what I discussed about the progression of Alzheimer's. I hold back from saying onset. The observation that the tau protein becomes heavily phosphorylized, and that leads to then the fibrillation of that protein, or excuse me, the formation of these this mass of protein. And so he was wondering if this can be any correlation with the use of lithium. And so I would say it's complicated, because as even you saw in this article, the lithium wasn't necessarily affecting phosphorylation, because you saw that it was affecting the levels of phosphorylation on some proteins or enzymes versus others. So some were becoming phosphorylated, some were becoming not. So it isn't a very clear thing that it's helping to, let's say, cleave phosphate groups or helping to add phosphate groups. It's something that there's something more fundamental or something more regulatory than that. And I don't think, at least in my opinion, that they necessarily got to the heart of exactly how the lithium is inducing that effect. What in that article, what they got to was to identify a protein that when it became phosphorylated, did appear to have an impact in terms of the response to lithium. So that CRMP protein, but it becomes complicated to translate treatment from lithium of that condition to then go into that neurodegeneration work. They're related, but it's a hard one to answer. All right, then. So with that in mind, we're going to transition to a discussion about the use of vanadium in terms of diabetes treatment. But you're going to see, especially when we talk about the in our seminar for Thursday, these themes about phosphorylation become still very important. And you will see today how phosphorylation also is critical to what impact vanadium may have as far as its therapeutic effect. All right, so let me go ahead and get that presentation. All right. All right. I probably have extra slides here that we'll be able to get through, but I want to get to the heart of this so that you guys have a context as far as metals and their potential for being used for anti-diabetes type 2 treatment. So part of this lecture was developed in collaboration with a former student of mine who is now a doctor, PhD professor in Columbia, Sergio Loza. And so we worked together in development of the material here, and also some of this material ended up in the manuscript that was published last year. All right, so as an introduction, when you're really talking about diabetes, you're talking about glucose homeostasis. And so here I just want to give you a perspective of the idea of regulating blood glucose and what would be considered a homeostatic level of glucose in our blood. So what would be considered a normal amount would be anywhere from 4 to 6 millimolar. So let's say 5 millimolar approximately. And so in terms of glucose getting into our blood, principal route is through diet. And so the glucose that can enter through our diet, part of it can come through the process of glycogenolysis, right? So the breaking down of polymeric form of sugar into its monomer, or even from gluconeogenesis, that is the development of glucose from these not necessarily sugars, but being biotransformed into producing glucose. Then when you achieve that level of glucose in your blood, and then what happens to the biological fate of that glucose in terms of its trafficking, right? Then some amount of it is going to get used, glycolysis, right? Some amount of it gets stored. So we talked about this the generation of glycogen, so this idea of making the polymeric form of the glucose, and then lipogenesis, this idea of converting the glucose into other material that your body needs. So here I give you an overview, this cartoon representation of, again, this idea of homeostasis. So this is more of a visual of what I basically mentioned. So imagine diet being our intake of sugar because of the whatever sweets we have in our food, or maybe some carbohydrates that can be converted into the sugar. You'll get an increase in the blood sugar. And so the way your body responds to this intake, assuming a healthy body responds to the intake of blood sugar, is that your pancreatic beta cells will release insulin. And so that insulin is actually really important because it serves as a signaling agent. For instance, it will go to your adipose cells, and that basically will signal that there is glucose present, and that leads to the uptake of the glucose into those cells. As a consequence of the uptake of the glucose into the cells, you're going to have a decrease in your blood sugar. And so if you're thinking about other factors that can lead to a decrease of blood sugar, obviously exercise can do so. So then what happens when you start falling low in your sugar levels? Well, then you're using the sugars. Your pancreatic alpha cells will release a peptide, a hormone peptide, glucagon. And so then glucagon, what it does, enables the breakdown in the liver of glycogen, so the polymeric form of glucose, into forming more glucose to reestablish homeostasis. So here we just kind of get an overview of the molecular mechanisms that are in place to control for intake, and then to control for when you start to fall below the optimal levels of sugar itself. Here I provide you a table, just a list of 5. There are more glucose transporters, and so what you get here is a list of the tissue distribution and what specific function they may play. So some of these glucose transporters help to deal with the uptake of basal amounts of glucose, whereas others help to deal with regulation of glucose and other sugars, particular ones, right? So you just get an overview of where these transporters are located and what their particular function is. But when you're talking about sugar, then, and I already mentioned, insulin plays a really key role in terms of maintaining that homeostatic control of sugar. So let's talk a bit about what insulin is. So insulin technically is a protein. You do hear language about it being a peptide hormone. And so why it's sometimes referred to as a peptide is because in terms of its structure, you can think about it as far as the a really small peptide interacting with other monomers of itself. What you can see here, just some of the properties, consist of 51 amino acids. And so those amino acids compartmentalize into 2 different chains. You have chain A, which in this depiction here, you can see it consists of about 21 amino acids. That interacts with chain B, which then consists of the other 30 amino acids. And so, as you can see, the interaction, at least one of the things that are enabling chain A and chain B to interact, are these disulfide bonds. Okay. Now, so that gives us a perspective of what would be the monomeric form of insulin, of those 51 amino acids. It turns out that the alpha chain, the beta chain, they themselves can dimerize, but this protein can adopt even higher order structures. So produced and stored in the body as a hexamer. And so I have a picture of the hexamer over here. And so this hexamer actually takes advantage of coordination chemistry, is stabilized by 2 zinc 2+ ions for high long-term stability. All right, so what are some of the functions? Well, control cellular uptake of glucose in muscle and adipose tissue, increase of DNA replication and protein synthesis via control of amino acid uptake, and also stimulates glycogen formation in liver. So here I give a little bit of the structural details. So what I'm showing here is the beta chain and how the beta chain interacts with itself, and it's stabilized by hydrogen bonds. When we look at the monomer, okay, of the alpha chain, the beta chain, there is a very important amino acid, the histidine there, which serves as a metal coordination site. So histidine, start thinking about hard, soft, acid-base theory. You probably guys will get bored of me saying this a lot, but it's a repeating theme, so I will emphasize it. So that actually plays a very significant role in terms of the interaction of these monomers, but also the dimer of the beta chains, to facilitate the formation of this hexamer that you can see here. And so that hexamer is stabilized by the presence of zinc. And so here you can see that there are 2 zincs. What I'm showing you is how 1 zinc is coordinated in this picture right here, and is coordinated by the corresponding histidine 10 of a corresponding monomer. So with each dimer, right, you're going to have then 2 zinc coordination sites, each consisting of what basically would be a trigonal coordination of the zinc itself. All right. So this form of the hexamer serves as a very stabilizing form of the protein, but it's not necessarily the form of the protein that is the most useful for us. It's actually the monomeric form of the protein is most useful in terms of facilitating signaling to whenever there's an intake of glucose. So oftentimes, as far as like the use of insulin, the formulations tries to favor the use of monomers just because of the of its ability to help in terms of the being able to deal with glucose intake into sugar. All right, so let's talk a bit about this process of insulin secretion in response to glucose uptake. All right, so here we have a depiction of your human pancreas, and then within there you have your islet of Langerhans. So the hundreds of thousands of cell clusters called islets of Langerhans where beta cells live. The beta cells release insulin in response to increased blood glucose levels in 10 minutes. Any alteration in the beta cell functioning has a profound impact on glucose homeostasis. It can lead to an excess secretion of the insulin, so hypoglycemia, or it can lead to an insufficient secretion, the opposite, and this would be type 1 diabetes, or characteristic of type 1 diabetes. All right, so let's talk a bit about, well, what exactly is the hormone role, the signaling role that the insulin is playing. So what we see here is a cartoon representation of insulin interacting with its receptor at the membrane, okay? And so we're looking at that point of interaction with the membrane and ultimately what impact it has with these transporters, the glucose transporter 4. So we're gonna see that correlation at the end of discussion of this slide. All right, so the first thing is that insulin binding, basically what it does, it activates the insulin receptor into a tyrosine kinase. All right, so kinase, I mentioned that last time, right? This process of phosphorylation. So basically now you have this active protein, or in this case is being referred to as an enzyme because of this kinase functionality. What does it do? Well, then it starts to then phosphorylate other proteins. So the enzyme phosphorylates itself, leading to the binding and phosphorylation of the cytosolic protein IRS1. What that then leads to is a phosphorylation cascade. So you get a series of proteins that are getting phosphorylated. So that's the cascade part. And that phosphorylation process serves as a signaling process that out here somewhere in the extracellular space, there is glucose. And so now we got to prime the glucose transporter to translocate from the cytosol to the membrane because we have to, intake that glucose. So that is basically the signaling that the insulin is doing. So now this particular transporter, the glucose transporter 4, is able to facilitate that uptake of the glucose. But then when you lose that control, again, of the glucose homeostasis, but also very importantly of the insulin itself, what it leads to is a really detrimental disease known as diabetes mellitus. So this is a chronic disease that occurs when the pancreas does not produce enough insulin or when the body cannot effectively use the insulin it produces. This leads to an overproduction of glucose in the liver and underutilization of glucose by other organs. So this disease is characterized by persistent elevation in blood glucose levels. You have levels that exceed the 5 millimolar that I mentioned to you previously, even when you haven't eaten, so even after fasting. So uncontrolled hyperglycemia will lead to serious multisystemic damage, especially in the nerves and blood vessels. The diabetes type 1, the typical association is that it's deficient to no insulin production and requires daily administration of insulin, something that is not preventable, this particular disease. Type 2 is when the body's ineffective use of insulin is largely, or at least this is a perspective that people have, is largely the result of excess body weight and physical inactivity. It's similar but less marked to type 1, but may be diagnosed several years after onset once complications have already set in. But I will say there are studies to suggest that this disease is even more complex than simply type 1, type 2, and I've even seen some suggested of a gestational type of diabetes. So in this study, they divided patients that were observed with diabetes into 5 different clusters, of which some of these clusters are characteristics of type 1 diabetes and some are characteristics of type 2. So the way I have written this is as reported in the manuscript. So the language is a little bit as more of an observation. So for instance, cluster 1, this is a severe a severe autoimmune diabetes. It's broadly the same as classical type 1. It hit people when they were young and seemingly healthy, but an immune disease left them unable to produce insulin. Right? So this is something that was outside of their control in terms of a lifestyle decision that may have led to this. It's something to do with their actual immune. Whereas cluster 2 is a severe insulin deficient diabetes patients that look similar to cluster 1, okay? But this is not something that was immune induced. And then we get into the clusters 3, 4, and 5, which more characteristic of type 2. And so here, for instance, cluster 3, you have the severe insulin resistant diabetes, were generally overweight and making insulin, but their body was no longer responding to it. Versus, let's say, cluster 5, which appears to be age-related, right? And but produce an onset of a mild form of the disease. So here, there's an added complexity of things that are due to lifestyle, let's say, excessive eating leading to being overweight, or due to simply age leading to an onset of the disease. So that might have something to do with something more potentially genetic. And then versus class 1 possibly being due to an autoimmune disease. So this study here that helped to try to get a more, I would say, more clarity about the onset or the effect of this disease or potential onset. But I would also say that it contributes to a greater complexity of what this disease is. All right, but in general, what are the common consequences of diabetes? So it can lead to damage of the heart, blood vessels, eyes, kidneys, nerves. Adults with this disease have 2 to 3-fold increased risk of heart attacks and strokes. Combined with reduced blood flow, neuropathy in the feet increases the chance of foot ulcers, infection, and the eventual need for limb amputation. A major consequence of a long term of this disease is diabetic retinopathy, an important cause of blindness. 2.6% of global blindness can be attributed to the disease. And then, as many know, it can lead to kidney failure and then to the need for dialysis. So here I found some statistics. I would say this is older, but still relevant in terms of the abundance of the disease. So 422 million adults have diabetes, 3.7 million deaths due to diabetes and high blood glucose, 1.5 million deaths caused by diabetes. So here are some additional statistics with regards to the number of people and how much it's progressed over the years in its general prevalence. What I want to show you guys is something that's more current. So I obtained this data that corresponds to 2019 versus 2000 so that we can see the leading causes of global death, right? So the highest one is heart disease, and then what we can find is diabetes at number 9 here. But you can see how from 2000 to 2019, it's progressing. You can see where Alzheimer's is here, something we were talking about previously. When we bring this into perspective for Puerto Rico, and I couldn't find something more recent than 2015. So if anyone can find something better, please share with me. So you can see here, causes of death in Puerto Rico. The number 1 is tumors. And then you can see where diabetes falls. Diabetes falls 3rd. And then below that, you can see the Alzheimer's. Okay, so let's talk a bit about, let me see. I have a question here. Daniela, why does the body stop responding to insulin diabetes caused by lifestyle? So basically the effect of, let's say, just eating crappy food, too much excessive sugar can lead to the body not being able to regulate all that sugar. And so if you have such a large excess over time, you're going to have, you're going to disrupt your metabolic process. But I'm going to talk a bit about what leads to, or at least what's attributed to the insulin resistance, the type 2 diabetes. So what I'm showing you here is the effect that is attributed to this protein, this enzyme, protein tyrosine phosphatase 1B. Okay, remember phosphatase, so the removal of phosphate. So it appears that it shuts down insulin signaling by acting at the insulin receptor and downstream signaling components, such as IRS1, leading to its dissociation from the insulin receptor. So basically, this enzyme appears to affect that interaction between the insulin its receptor and then the subsequent phosphorylation cascade. So what we have here is a representation of the receptor, the tyrosine has a phosphate group there. And so what we're seeing is how that substrate is positioned with respect to a very critical cysteine group at the enzyme active site. And then you also see the enrollment of aspartame and arginine in terms of positioning of the substrate. So I'm going to clean this up so we can look at this with more clarity. Let's see what's actually happening. So we have that phosphorylated tyrosine entering the active site of the PTP1B, where there's a very critical cysteine group. And as you can see, that cysteine is serving as a nucleophile. And what it's ultimately doing, it's interacting with that phosphate on the tyrosine, such that now your substrate becomes dephosphorylated and your cysteine is bound to that phosphate group. So you can see that right here. So you have that phosphate group, then you have a rearrangement of the active site being facilitated by this glutamate as well, which will lead to the dissociation of the phosphate, and now you're basically regenerating your enzyme. You're regenerating that free cysteine, which can then go on to act with additional substrates. So what that enzyme is doing is it's leading to very critical dephosphorylation events, and apparently you have an overproduction or overactivity of the PTP1B that's affecting the phosphorylation of the insulin receptor and downstream of that, which then affects the sensitivity to the insulin and the signaling from insulin. All right, so here's just a summary of what I just said with regards to what that cysteine of the active site of PTP1B is doing with respect to the phosphorylation. All right, so Ladimaris has a question here. So she says, en la misma línea de que hay personas que hacen diabéticos para llevar una buena alimentación y en ese caso que lo causan. Right, so I already gave you a list of possible onsets of it that may have nothing to do with lifestyle. So it could be an autoimmune, it could be other factors that as simple as age and whatever corresponding ailments to do with age, because there could be quite a bit there. All right, well, it was observed that vanadium compounds or vanadium species can exhibit an anti-diabetic effect. And so the French physician Lionnet, who was the first person to report that this vanadate species could potentially be used to treat diabetes mellitus in 1899. But it was in the 1980s that really the anti-diabetic effect of vanadium salts were being explored. So here were some of the initial salts, and I say salts versus vanadium complexes, right? Because these are just vanadium, in, let's say, potentially in an aqueous environment, this would be what would form. These are not chelators or even ligands, external ligands, they are bound to the vanadium itself. So working with these salts, and then we're working here with vanadium 4, which is D1, EPR active, vanadium 5, which is D0, not EPR active because it's not paramagnetic, but it actually is interestingly vanadium 51 NMR active. So you can actually study this metal by that tool. Just to give you a sense of how you can study vanadium 4 and vanadium 5 using spectroscopy. All right, so what was observed? It was observed that vanadium salts in drinking water resulted in normal glycemia in diabetic rats, and that the vanadium salts mimic most of the physiological effects of insulin. Right, so here are some of the representations of what was observed: stimulation of glucose uptake in fat cells, enhancement of glycogenesis, stimulation of fatty acid formation, reduction of cardiac decline, and streptozotocin diabetic rats. Basically, when you're using the streptozotocin, what you're doing is you're killing or you're affecting the health of beta cells, and so you're compromising them. They're not able to release the insulin, so that you're essentially inducing a type of diabetes. But the big question was, are the vanadium salts, are they insulin mimetic or insulin enhancing? And I would say, even kind of frustratingly, this question still exists today. I had a review article that someone insists on one being true over the other, but whatever. So the first thing was that, is it insulin mimetic? Vanadium was believed to replace absent insulin in diabetic rats. So a study was done to probe this. There's bio-breeding diabetes-prone rats. Vanadium was not enough in the study to keep rats alive in total absence of insulin. But what was observed was that if you did have insulin, you treated them, they were able to exhibit normal glycemia. If you had some insulin, but not as much as the other study, and then vanadium, you were able to achieve normal glycemia. This experiment and other similar ones led people to suggest that vanadium 4, vanadium 5 is not an insulin mimetic, but rather something that enhances sensitivity to insulin. All right. And I would say I fall into this camp of thinking. But maybe in the future, I'll be proved wrong and I will have to change my slides. Although I changed my slides quite a bit. Anyways, so I want to bring us back to our discussion about speciation of vanadium 4 and vanadium 5, as it was a subject of your assignment to review this information that I presented to you. And very interestingly, when you look back at the speciation of vanadium 4, you find something really important, that in this pH range of, say, about here to here, you have this species, this vanadium 4 species, looks like this, and it actually is comparable to phosphate, the structure of the tetrahedral structure of phosphate. And so the suggestion is that, well, perhaps the vanadium 4 itself, that ion that is produced, this anionic species, well, maybe it's serving as an inhibitor of phosphatases because it basically appears like phosphate. So maybe it's mimicking phosphate, and that's its functionality. So people explore that possibility by looking at vanadium 4 inhibition of phosphatases. Okay, and here's an example of a structure with the vanadium binding to the cysteine group. Remember the cysteine of the active site of this enzyme. And so some things were done to try to characterize this. For instance, a measurement of the dissociation constant. And as it turns out that the vanadate, the vanadium 4, actually has a higher binding constant or lower dissociation constant than phosphate. And so if you look at this structure here, the type of structure that you can see here is 5 to 6 coordinated, highly stable complex with the phosphatase active site cysteine residue. So the vanadate appears to be inhibiting the enzyme by not enabling it to bind to the phosphate. A similar analysis was done for vanadium 5. Remember, vanadium 5, the speciation is much more complex. If you look, though, in this range, and actually it starts up pretty higher up, you see this vanadate vanadium 5 species, and that is actually isostructural and isoelectronic with phosphate. And so there has been suggestions that vanadium 5 can play a similar role as vanadium 4, but I would say a lot of the work has been more focused on vanadium 4 in terms of its inhibition property, especially because of the formation of that species is for the vanadium 4 is more likely in the physiological range, whereas for vanadium 5, you actually have to go up to higher pH range, above pH 8, to see any significant amount of it. All right, so then that really, altered the field, and so people were really going into and trying to develop vanadium for compounds and also for oral administration. So here were two really important findings. The bis modulato, so the modulato ligand, oxovanadium 4, and the bis ethyl modulato oxovanadium 5. These two compounds. And people were really interested in them because these compounds follow the Lipinski's rule for compounds that would potentially be orally active. And then in terms of structure, you have this square pyramidal type structure. All right, so what was observed with these compounds? They're small molecules. Well, then you have these CHO cells that inhibited overexpress PTP1B. So you did see an effect of these compounds on the enzyme. The male antidiabetic Wistar rats, oral administration or injection resulted in normal glycemia for 2 days. And then in these male diabetic mice, this mice model, when you inject this dosage of these compounds, normal glycemia was observed in 18 hours. So clearly in these rat model and these mice diabetes model, there was an impact of these compounds. And so here's a representation of what the data would look like. So you basically have to do a blood measurement to determine how much glucose is in the blood. So you're looking at plasma glucose versus time, okay? And then what happens in the presence of these two compounds. So you can see at you deliver that the compound at this dosage, what's happening to the glucose levels? Well, you're starting off at 20 millimolar. That's 4 times higher than what I said to you before, right? But then over time, let's say at one day, you get down to about, I would say maybe 7 millimolar. So you're getting down to a level that is actually considered healthy. And so you can see over here in this other plot, plasma glucose versus dosage, right? And then you have to get to this dosage here to see what would be normal glycemia. I think even 0.6 might be sufficient for that as well. Okay. All right. So the this version of the compound, the BEOV, it actually advanced to clinical trials. It actually made it to phase 2 clinical trial. There's actually kind of a sad story as far as the people that really pushed this forward, and essentially a little bit of a a sad door closing event for the field. So here, this is a dosage that was administered, but unfortunately, there was simply not sufficient efficacy observed, so this compound was not advanced any further. But I would tell you that there was a lot of work, time and effort put into really try and push forward a vanadium compound for this purpose, and basically it stopped here. And the people that were really leading this have since stopped pushing this particular research forward. But to try to get an understanding of why that happened, well, you can look at aqueous speciation. It can give you some answers. So if you look at the Modulato species, right, and you think about the oral digestion, so you have to go through your gastric tract, well, you're in this range, acidic range. And in that acidic range, what happens to that compound? Well, according to this, the biological fate is that at pH 2, you're basically going to have a dissociated compound. So you're just going to have free vanadium 4, okay? And I know it says VO2+, that's vanadium, but the vanadium itself is vanadium 4. And that form of the metal simply will not be absorbed sufficiently by the body, and it will just get excreted out. So the delivery mechanism could potentially lead to just the compound not being effective enough. But even if you look at higher pH values, right, you do have an intact compound in I would say the more physiological range. Your pH 7 is pH 7.4 pH of the blood. So you do have the compound intact. But delivery through the oral system, you're going to have a significant loss of nearly, basically 80% of the compound. And so a very small absorption. So in addition to this, people want to understand, well, what is the potential fate of that vanadium? And we talked a little bit about this when we talked about speciation of vanadium. Well, what they try to examine, well, if that vanadium compound makes it into the blood, what happens next? Well, what happens next is believed to an extent is that the compound can interact with the serum transferrin protein. And here we have 2 models representing how the vanadium may be able to associate at the metal binding site of this protein. Remember, this protein I talked to you guys about is an iron binding protein. Well, here in this one example, you have the vanadium interacting with some of the protein residues, but you also have one of the Modulato ligands there. And that Modulato ligand, like the carbonate ligand, is serving as a synergistic ligand to help stabilize the vanadium. So here you have basically part of that small molecule binding to the metal binding site of the protein and then potentially being brought into the cells via the transferrin receptor. We also have another model here where the actual intact compound binds to a surface site of the protein. Okay, it doesn't actually, in this model, the vanadium is not bound at the metal binding site. But being bound by the surface site of the protein, if this protein is iron bound, then there's another mechanism in which that compound can enter into the cell, because then the iron-bound protein is recognized by the receptor, and then you have the endocytosis that's potentially induced. All right, it's not clear which of these mechanisms really is the preferred mechanism, but these are 2 possibilities. So putting this all into perspective, well, what is the biological fate of the vanadium? So again, through the oral mechanism, you have a significant amount that's just simply going to get excreted as the vanadium ion, and a very small amount absorbed into the body. But then if there is some amount that gets absorbed to the body, some of it gets affiliated with transferrin, and then you get potential biodistribution. if you were to go through a different route, bypassing the oral delivery and either an intravenous or intraperitoneal delivery, well, then you have probably a higher amount of vanadium that can get biodistributed, that can potentially enter into the cellular environment. And so within the cellular environment, well, what actually is potentially happening? So again, the data indicates that the vanadium may potentially then get delivered to transferrin, or depending on some other mechanism, maybe you have some free amount of the vanadate itself. But vanadium affiliated transferrin gets brought into the cellular environment, and then what happens next? Well, then that vanadium, the free vanadium anionic species, has a capacity, or the vanadate species, depending on how you want to look at this, has a capacity to bind to the cysteine of PTP1B. And in binding the cysteine, you effectively inhibit it, and that then will prevent that enzyme, the PTP1B, from cleaving the phosphate groups of the insulin receptor. And so, in theory, then, you would be able to facilitate the phosphorylation cascade, enabling the glucose translocation, the intake of glucose. In other words, you're able to facilitate the sensitivity to the insulin that binds to the insulin receptor. Okay, so that is what people have proposed to be the mechanism of the vanadium, that it serves as a phosphate mimic able to inhibit the cysteine of the active site of the phosphatase. And so a little bit of looking back at some stuff that I already covered before, remember I said to you how many of these small vanadium compounds, when you study them in terms of the speciation at a pH 7.4, the pH of blood, many of them will dissociate and form either vanadium transferrin compounds, or maybe vanadium compounds with other bioligands, right? But if we look at this one molecule with the picolinate, okay, and that was actually done in a subsequent study. This is a representation of the vanadyl picolinate complex with two equivalents of picolinate. Well, an actual study was done with EPR, because the vanadium-4 is EPR active. And so it was observed after intravenous administration of the compound, and then they were tracking the blood into a system that was connected to an EPR instrument. And here is the data collected of the EPR scanned over time. And so this is the data collected over time, the signal. And what was observed was that there were at least 2 EPR active species, 2 vanadium-4 species observed over this time frame. And what this also suggested was that the vanadium picolinate complexes are actually present. So at further studies, not just from this blood analysis, but also then processing the tissue of the mice that were examined, the vanadium picolinate complexes were present in the treated organs. Bone and other tissue have the capacity to incorporate vanadium, and they do so by binding the metal as a complex. But it isn't clear how this binding occurs. So the vanadium species that clear from the blood are believed to redistribute into the tissues before ultimate elimination in urine. So what this data suggested was that even though the aqueous speciation study suggests that vanadium picolinate should basically dissociate, this study shows that in vivo is much more complex than that, and a good amount of that vanadium picolinate through intravenous delivery actually did biodistribute much more than expected. All right, so that's why I'm gonna leave you guys with, well, as a transition to our next class, which will focus more on anti-diabetes from the perspective of chromium 3+. All right, everyone, I'll see you next time. Take care.
Thank you.
You're welcome.
Professor, can I ask you something about the
Go ahead.
assignment? Okay, so I have about the second assignment, the first question. I have doubt with that one, like you told me que estaba como que no era eso lo que se supone que respondiera, y pues tengo dudas.